Okay, thank you so much. And I'm going to be talking to you about my research on West Nile virus in New York and Connecticut, where we were finding importance of temperature and soil moisture. And I know for this group, it's a vector control group, and so I apologize in advance. My talk is probably going to tend a little bit more towards the academic, but I do appreciate you being here and talking and hearing it because from the academic side, as Emily mentioned in her introduction, we're very interested in trying to be able to bridge the gap between what's happening in academia and being able to apply this to modelers. And so I'll talk a little, or I mean, not modelers, but to the vector control. And so I'll talk about a little bit about those efforts at the very end of things. And I do just want to mention I'm at University of Albany. I'm also through the Department of Health. And we've been collaborating with the Connecticut Agricultural Experimental Station as part of the Northeast Center of Excellence for Vector-Borne Diseases. So many of you are probably very familiar with West Nile. I don't have to spend too much time on this, but over 50,000 diagnosed cases in the US, over 2,000 deaths. In the study area that we're looking at about 1,000 cases in New York and around a little over 150 cases in Connecticut. We chose a Northeast focus for the study because this is where West Nile was first introduced to the USA, was via New York City. West Nile is most of the deaths are associated with neuroinvasive West Nile. It's where it invades the brain. And the incidence of neuroinvasive West Nile increases as you get older. So at older individuals, higher risk of getting the neuroinvasive West Nile. And it's worth noting that of those 50,000 cases, the vast majority of cases of West Nile go undetected. Only about 20% result in any symptoms. And it's only, I think, less than 1% of them that actually become neuroinvasive. And so this risk of actually getting the severe disease increases the older you are per 100,000. It's also a very seasonal disease. So this is for the United States. It's only going through 2016, but it starts to you start to see it into end of July, into August, peaking kind of mid-August, September, declining into October, but still getting some cases into November. And this is a very interesting pattern in that this is for the entire US. So that includes the southeastern US, includes the western US, includes the northeast. And so across all of these locations, we're still seeing the seasonal pattern in human cases of West Nile. And so that's kind of a little bit of a strange thing. It might be tied in some ways to the bird populations, that if you have a pulse of birds migrating back to the US or breeding and the breeding productivity, there could be a relationship there of the new naive hosts for the amplification. Because as many of you probably know, West Nile is primarily a bird and mosquito disease. And so it's the mosquitoes that are getting sick with it and the birds that are getting sick. Well, I don't know that the mosquitoes really feel any symptoms from it, but birds getting sick with it. And humans and horses, which are what a lot of the public health departments care about, are dead-end hosts, so they're not part of the amplification cycle for West Nile. So the avian impact it's worth thinking about is that the impact on birds of West Nile in the US has actually been larger than the impact on humans. Over 332 species have tested positive for West Nile. This includes some zoo animals, but this is just from the United States. It's been responsible for the deaths of millions of songbirds, although some of the ecological impacts of that haven't been that long. So I mean, one of the studies was estimating there was probably 29 million red-eyed vireos, a type of small bird that died. But on the other hand, the next year, the populations were back to normal. So some of these birds can respond very quickly to some of these populations. More noticeable was about the 45% decline in American crows, and American crow populations had been about around 80 million. And so at the peak, you're talking about tens of millions of dead crows possibly from West Nile. So our research in our lab has been focused on climate change. Our research group is really focused on climate change and emerging infectious diseases. And so the thinking about how would climate affect West Nile? And one possibility is when the climate conditions favor the increased mosquito populations. Another possibility is when it changes the interaction dynamic between the mosquitoes and the birds, potentially through changes in the bird's phenology, if the birds are coming back earlier, if they're staying around longer, if they switch from being migratory to being resident. That could change the West Nile dynamic. Climate conditions can also affect mosquito interactions with humans. So that spillover aspect might be climate dependent. If people are staying inside in the air conditioning because it's very hot out, they might not be in the same position where they could be getting bit by the mosquitoes. In contrast to for this woman here, if you have a really nice day, people might want to be out in their garden, working on their garden. It might increase their risk of being bitten by mosquitoes. It can also have a direct effect on the replication rate of the virus. This is primarily going to be seen in the mosquito side of things because of the virus inside the birds maintain their body temperature at a constant temperature. But on the mosquitoes, as the temperatures get warmer, you typically have a faster replication of the virus, and the mosquitoes can become infectious and spread the virus faster with an increased temperature, although there are upper temperature limits. There's some really great research coming out recently looking at the upper temperature limits on West Nile. So some of the broad patterns in the literature, though, is that typically you see an increase in West Nile with increasing temperature. And this has been shown for winter temperatures, spring temperatures, and also summer temperatures. Results have been more mixed with precipitation. So some studies have shown no effect of precipitation. Other studies have shown an increase with increasing drought. And other studies have shown an increase with increased precipitation. And I've got some results in here that I think really help illustrate what might be going on with the precipitation. So bear with me. The question then is, can West Nile virus be predicted? And this is what we're really trying to work on. There have been a number of studies that have been looking at this. So Nick DeFelice has a real-time model that takes incoming surveillance data and then processes it and makes predictions for what the West Nile season is going to look like. There's some studies on Suffolk Island using a statistical model. The Menor study was across the entire US. And these studies have varied in the spatial and temporal scales at which they're making predictions, the results, assessments, metrics that they've been using, and which variables get included. And so this can make comparing across studies a little bit messy. And we'll talk a little bit about that further on. In our study, we were trying to test a specific hypothesis of do these climate variables improve the prediction of West Nile for human cases or mosquito infection rates if we include other covariates? And if so, which climate variables are important? Also, are these predictions improved by using a finer spatial scale? So we ran our analyses at the county scale, which is what the modelers like to use because you can aggregate the trap data across all of the traps, and it gives you neater numbers. It makes it easier to make the predictions. But on the other hand, it also, when we've been talking to vector control, they also say, but having a county level prediction, how does this help us? We want to know where we need to spray. We need to know where we want to do interventions. And we only have the budget to target that at very local areas. And so we were looking at trying to see if we can find this balance between predictability and applicability. And the spoiler alert is we're still working on that balance. But it's definitely in we're trying to work on that and trying to think about it and figure out how to make these results more useful to vector control. So for this study, we were using data from New York and Connecticut from 2000 to 2015. We were only using data from gravid traps on the basis that the mosquitoes in the gravid traps could potentially be infected with West Nile. We were looking at Culex pipiens, Restuans, and Salinarius. So the Culex mosquitoes are the primary vectors, both for amplification and probably also for bridging West Nile to humans. Salinarius probably more as a bridge vector, and Restuans probably more as an amplification vector, and pipiens doing both. We were looking at mosquito infection rate and number of human cases, and we were aggregating our climate data from January to March, April to June, July to September, and October to December. So we looked at temperature variables, growing degree days, so when the cumulative temperature was above 10 Celsius. We looked at minimum and maximum temperatures for that quarter, mean across the quarter. And then we also looked at extreme minimum and extreme maximum temperatures for the quarter, thinking that maybe some individual event might be most important. Spoiler alert, it's not. The extreme variables did not come out in our final models. We also looked at moisture variables. So we were looking at precipitation and precipitation intensity, and also soil moisture anomaly. And we also looked at deviations from normal for the climate variables on the basis that maybe it's the deviation from normal that's more important than the absolute values of the variables. We looked at a variety of non-climate variables. Because we're short on time, I'm going to gloss over these, but they had to do with mosquito abundances, human populations, land cover, and some details looking at the birds, because some birds are, like the robin is a very good spreader of West Nile. If a robin gets infected, it's very easy to infect other mosquitoes, whereas a mourning dove might not be able to infect another mosquito if it gets bit. And so birds are varying in how infectious they could be to a mosquito, another mosquito. And so we were trying to combine that with a host competence index. In the final model, none of the bird variables came out into the final model, but we were at least looking at it, trying to control for that. So statistically, we were looking at mosquito infection rates and human cases, and we were using a random forest approach. And I'll talk a little bit more about that in a moment. So we first screened all of the different predictor variables that we were looking at for our model. And then we reran it with just the important variables. The importance is determined by taking a predictor variable. So say it was the growing degree days. What the statistics does is it jumbles up all the values and then puts it back into the model. And if you get the same answer, that variable is not important. And if you get a very different result in the end, that was a very important variable. And then we did another fitting process where we took out any variables that didn't have any unique explanatory power to try to give us a minimum subset model with just the key variables for prediction. Now, these might not be the mechanistic variables if things are correlated, but it gives us a smaller, reduced model. And then we evaluated our models by leaving a year's worth of the data out. We refit the model and then tried to predict the missing year's data from the refit model to really get a sense of how the model was performing without an individual year with the key variables. So random forest, you take climate data, non-climate data, and West Nile virus data, you put it into the black box and you get the answer, right? Well, I want to try to open up that black box and not just have it be a black box, because that's honestly, from what we've been talking to vector control, that's been one of the barriers to using some of these more advanced statistical models, is that they don't want to see a model that they can't understand. They want to have some intuitive idea of what the model is actually doing. And that's one of the things I like about the random forest model is even though it's a machine learning approach, it's still something that I can understand. And the first thing I want to say when I'm talking about random forests, there are no living trees involved. Well, some might have been used to print the original press article, but what we're talking about is, instead of living trees, we're talking about cartographic regression trees. And so it's a statistical approach. And it's actually a very simple statistical approach. What it basically does is, if you take your data that say zeros are absence of West Nile and ones are positive of West Nile, and the X is some variable of interest, you sort your data based on thresholds. And so in this case, it's very neat, it's tidy. You can say X less than 7, no West Nile. X greater than 7, you always get West Nile. If only it worked that way in the real world, it would be wonderful. I probably wouldn't have a job because somebody would have figured it out 2 decades ago. But the random forest approach, the real world is messier. It's a more challenging situation, and the random forest approach accommodates this messiness by saying, if we actually use subsets of this data and we do this splitting the process a whole bunch of times, and you can have more than just one variable, as in this cartoon example, and we build out these regression trees, we actually get a better result than if we just do it once with the cartographic regression tree. And so that's the basic idea here, is that you're splitting data based on thresholds. And you're doing it a large number of times to try to get some sort of replication and to be able to average over a large number of different scenarios, and to not have your results be skewed by a few particular values. And the studies have shown that it works better. And so that's what we went with our approach. So the strengths are, it's a machine learning technique. You can capture nonlinear responses. We can describe complex responses. You can screen a large number of variables, and the method can still be intuitively understood. So here is some of the data from just some of the counties that we're looking at. So you can see that they're varying in mean minimum temperature. So Nassau County is our warmest county, and Erie was one of the cooler counties in the study. You can see that the mosquito infection rates are varying substantially by year and by county, and also the numbers of human cases. So do the climate variables improve predictions? Yes, we were seeing 17 to 19% more variation explained with the mosquito infection rates, and 7 to 12% more variation explained for human cases. This is a quick, This is just another way of looking at the results. If the model was perfect, we have the observed mosquito infection rate on the x-axis, the predicted on the y. The model would have it all on the line. And the quadrants are just an arbitrary classification based on 5 infected mosquitoes per 1,000, and our model is 84.4% correct with this threshold. Now, it's worth noting that if you had a model that just always said that the West Nile is absent, you'd be right 72% of it. So keep that in mind when we're talking about accuracy. Is the big concern is that it seems to be we're only getting about half of the high West Nile years are being identified by the model. And so that's a little bit concerning. To try to visualize this is for mosquito infection rates for 2012. For convenience, we divided it up into blue is less than 1 case, red is greater than 5, or less than 1 infected mosquito per 1,000, red is greater than 5 infected mosquitoes per 1,000. And you can see that the model is both over-predicting and under-predicting in some places, but it's catching the general pattern. Similarly with human cases, there's some areas like the Hudson Valley that the model didn't do very well on. But you're getting some, but the model's catching the general pattern for New York, and Connecticut is right here, just north of Long. So we were finding that mean minimum temperature from July to September was the most consistent variable. This is a little bit discouraging because the CDC West Nile forecasting challenge wants the first forecasts at the end of April. And our most important variable with this method was coming out to be summer temperatures. And so that might suggest that there's some limits to predictability for what you can do in a forecasting challenge. If the important variable hasn't happened yet, it may make it very difficult to predict West Nile in a year back in April. And then selected variables differed for the human and mosquito analyses depending on the spatial scale. We saw soil moisture and temperature for mosquitoes at the county scale, but temperature only variables for the mosquitoes at the point scale. In the interests of time, so the contour plots we then tried to evaluate this, we tried to visualize the results. So we had one variable on the x-axis, another variable on the y-axis, and we had the model make predictions. We drew in contour lines and then plotted the observed values with the size scaled to the infection. And I really want to show you this figure because here on the x-axis, we have the soil moisture anomaly from April to June and the mean minimum temperature from July to September. And what we're finding here is this I'm on the wrong side. We're finding this U-shaped relationship, so that at low soil moisture, we're seeing a high risk of West Nile with these, you can see these larger circles. With this U-shaped pattern at high soil moisture, you also see a slight increase in risk for West Nile. And at high mean minimum temperature, you start to see a uniformly high risk of West Nile. And so what I like about this figure is it's showing that precipitation, all 3 of those precipitation results from the literature are being played out right here in this nonlinear response, where if you have drought, you have a high risk of West Nile, unless there's a high mean minimum temperature, in which case you always have you don't see an effective precipitation. And even at high precipitation, because of this pattern, you can see that same relationship. And so what we're actually looking at for precipitation isn't conflicting results, but a nonlinear response. We're going to skip this. So the important conclusions is that we were finding that these climate-related variables, especially temperature, were important in some spring and summer drought, were important for understanding the mosquitoes at the county scale. Some of these relationships can be very complex, and some of these environmental predictor variables can be very strongly correlated. So a little bit on our current and future directions. We're currently working on comparing this model to other models of West Nile being used in different regions of the US. And the goal is to really get a sense of what the different models can do, which ones don't work very well, and how do the models compare to each other? What questions, what decisions can the models be used? And so if you're interested in participating in this, if you have a data set or if you have a model, or you a vector control agency making decisions related to West Nile and you want to more incorporate West Nile into it, please drop me a line and let me know, because I'd love to hear from you and have people involved in this effort. And that's part of why I'm here today, is that we're really trying to get this comparison with as much data as possible, with as many models as possible, to really see if we can learn from the best of everybody in the US to really see if we can move the predictive modeling to a point where it's useful to vector control. Some of the other projects we're working on is switching more to a probabilistical forecasting framework than was just demonstrated here. And we're also doing some work on future risk of West Nile under climate change. And the short story there is it looks like it's probably going to be more geographically widespread in the Northeast. And so this is back onto that model comparison project. So we're looking at, it's a collaborative project across all 5 of the centers of excellence, the vector-borne centers of excellence. And we also have folks outside of the centers of excellence. So if you're outside of it, we're still happy to have you. And that's my contact information there. And with that, I'd like to acknowledge a whole host of people, especially the Northeast Center from which my funding is coming. And with that, I'm happy to take any questions if there are any questions. Thank you, and I apologize if that was a little bit rushed. I was trying to get us a little bit back onto time. So Ilya asks if we are planning to incorporate socioeconomic conditions into our analysis. The answer to that is we would like to, that's in the long-term plan. We haven't gotten there yet, but I think those would be great variables to add into the modeling effort. And Keith Price has asked, how would you factor in mosquito control efforts into predictive models? And this is an excellent question. So you'll notice it was missing from our study. It's missing from a lot of the models. The big challenge we've had with this is data. What we really need to be able to factor mosquito control efforts in would be to have data on what's going on for the mosquito control efforts. Because if we had those data sets, we could add it in as a variable. So you could say mosquito control, yes, no, in the scale, like with the year in county, you could say yes or no, or you could give some sort of quantitative measurement of it, and we could put it into the model that way. Another possibility would be shifting the model to a finer scale of resolution, and then you could directly model when the timing of those mosquito control events. But the problem is getting the high resolution, getting that mosquito control data. We've had a lot of trouble obtaining those data. The New York Department of Environmental Conservation keeps track of pesticide use, but when we tried to put it into the model, we really weren't getting very useful results. It was part of the problem was they had everything in there. And so trying to sift out the mosquito control from like the turf grass applications was really hard. So that's definitely something we would love to address in a more rigorous way. And so if you have mosquito control data and mosquito trap data and are interested in trying to get the modeling done, I'd love to hear from you.